She's mystified, has deep brown eyes. It's like a fire in my soul. And when she speaks, I get so weak. Oh, I'm losing my control. And when she smiles, a million miles, the stars in heaven start.
Hi everyone, how are you today? So this week's lecture is our final version of the lecture of the semester. And actually, I'm retaking this lecture recording again because I had the same problem of this file conversion crash. So it's been delayed more than I expected. My apology once again. And so with this, we should be able to finish the chapter 5, which is about the mutation, and within which we will be able to define basically what mutation is and pick up 3 specific examples of actual mutation cases. And the earlier, when I was recording the original version of this lecture yesterday, actually I had an extra time slot left after finishing this chapter 5, so I have included a little additional material of the next chapter, but I'm not really quite sure whether that's really necessary or not. So let me really think about it after finishing this part of this chapter 5, whether I should stick with the original the version of the lecture recording or not, because after all, considering that this semester I've got to include the additional chapter which is about the molecules, macromolecules, and etc., still I was able to accomplish this the up until chapter 5. So it was a little more than the amount of the coverage compared with the previous semester, 2 consecutive semesters. So I don't know actually. So let's get to it, and then we will cross the bridge when we have to. So having said that, mutation is defined as the change in DNA sequence, as I have mentioned briefly once again earlier. So mutation happens when DNA sequence changes, especially when DNA sequence on the like genes. Here the sequences that actually affecting the gene's function. So as we define the gene is actually I still didn't get the correct groove of this. So if this is a coding region, which will generate RNAs. Yeah, of course, there are several different kinds of RNAs. But also we do have this area, the regulatory region. So not only this actual the coding region of the gene, which correspond to exactly the area of RNA produce the RNA, but also when we consider gene, that area which does not appear on the part of the RNA, but still also essential in the making the gene function, is also the part of this territory which is affected by this DNA sequence change. So that should be also included in there. So anyway, overall, after DNA sequence altered, then as a consequence, the function or property of a gene has changed, then definitely we should consider that as an example of a mutation. What about somebody actually will say, then what about the DNA sequence is still, it is also possible that here's the DNA, you generate the RNA through transcription, so DNA is okay, there's no sequence change, but still, is there any way that RNA sequence change? So is a mutation. Yeah, technically, yeah, it is also a mutation. Obviously, in the more urgent and familiar cases of this coronavirus variant, the variant viruses are all the result of the change in DNA, the RNA sequence, because the coronavirus is an RNA virus. So it also counts, obviously. However, that's the kind of a different cases, because in the case of a coronavirus, their genome is intrinsically, their genomes are made of RNA. So that's a different case. In our case, this change in the RNA sequence usually is not considered as any legitimate mutation, because usually this is your original. When the RNA sequence change, that means it's just only one out of many, multiple copies, which means, yeah, you may have a one sequence changes, you usually generate ton of copies of RNAs out of one single region of the DNA, the gene area. So let's say like you have 2,000 copies of the same area of the gene, and only one or two or several of the 2,000 of RNA copies, the sequence has been altered, changed. It wouldn't really affect, it wouldn't determine any the consequences significantly. That's why we usually don't consider the RNA change as a part of the mutation. Yeah. So having said that, there are three different types of mutation can we think of. One is called the substitution, and another one is deletion and insertion. Basically, what it is, a substitution is all these can be displayed at the on a single point mutation level, like single nucleotide level. Single nucleotide level. These all three different kinds of changes can be displayed. Then we call that as a point mutation. Because like a single dot, the single nucleotide has changed, but still, depending on the actual the role of the single nucleotide in a gene overall scheme of the gene, it can have still a significant consequence as a point mutation. But also, all these three different types of alteration can also be played out at the level of many multiple whole segments. Yeah, the bigger scale of whole fragment level. So in the at larger scale can also, these three different types can happen. But anyway, by just looking at substitution, deletion, and insertion, you would know intuitively what they are. Substitution is something like a switching. You have this original nucleotide or some larger fragment of part of the your gene, then that's been replaced at the, like if single nucleotide and another nucleotide took the place of the original nucleotide. Like if the original nucleotide was A, for example, then another nucleotide of a different kind, like a G, has been substituted, like in that original place. That's what we call the substitution. Um, so one may think like, hey, it's a substitution. So even if it's just a different kind of a nucleotide, usually you have, on average in a gene you have so many number of nucleotide, like thousands or even more than 10,000, or you name. Then it's only one nucleotide. What kind of a consequence ultimately having this? You didn't lose anything. Would have. Yeah, usually that's the case, if that was the case. But sometimes, in a very rare, unfortunate occasion, that still will critically affect the outcome of this gene function. Deletion, in the case of deletion, is just the missing without any replacement is called the deletion. And an insertion is when you have one additional nucleotide or segment is inserted, added on to the original location of the part of the gene. So graphically speaking, there's been substitution is here. So here is the concept of a codon, as we have seen in the previous lecture. That's what it really makes the some kind of a serious consequence, potentially at least. So each nucleotide, here we are looking only the messenger RNA level because in order to illustrate the link between the codon and the result in the form of the amino acid. So this is an example of messenger RNA gene. So in messenger RNA you have 3 codon, each of which will signify specific amino acids. So at the point mutation level, if you had this, for example, base substitution, originally it was like here the G. The original nucleotide was G, but then it's been replaced by adenine. Then as a consequence, GGC versus AGC, which is totally different codons. So now the identity of the amino acid has been changed into something different. Then it can potentially make this greater impact in the function of the protein, that is which is important for us. Deletion, in the case of deletion, why deletion will also have some kind of different consequences. So now it will change the following the codons. So once you have this deleted, missing U originally you had, then now the following, the nucleotide will become a part of this previous codon. Then it is actually bigger disaster afterwards of this deletion. Okay. Same thing. The addition, the substitution is like usually something that only affect that one particular codon will be changed. However, in the case of a deletion, and then also you can imagine easily that it will also be the case in also the insertion too. So once you have this event, then not only is on that particular codon that is being altered, but the following From that point on, all other following nucleotides, the codon identity are all usually are going to be changed. Okay? This now with this type of a scenario, that's I'm going to introduce the concept of so-called the reading frame. So what we can say about this mutation among three different types of mutations, especially at the point mutation level, the single nucleotide change scale, point mutation, either deletion or insertion, then you have much greater impact on the product of this gene, usually in the protein coding gene, the messenger RNA coding gene, because this protein coding gene has this codon. Okay, three nucleotide will have one specific identity of one amino acid, then that will be disrupted. That kind of order we call as a reading frame. Okay. So that reading frame will be changed in this way. We call it as a frame shift. So once again, when we describe this type of scenario, what we can say about is a deletion or insertion of one single nucleotide, which is a point mutation, or even in some cases like two nucleotides. Still, it's can be considered as a point mutation because this is very small scale. So one or two nucleotide scales of deletion or insertion will definitely result in so-called this frame shift of what? The reading frame. The frame, what we could define as a reading frame, will be shifted. So it will be a mess. So then what's a reading frame? Reading frame is this, like three nucleotides, so that you will have this order of and the rule of reading three nucleotide by three nucleotide. So in a given area, in a specific area of the DNA sequence that's shown over here, actually depending on where you start reading this 3 by 3 by 3, you have potentially three different possible reading frames exist on the same region of the DNA sequence. If you start right at the first nucleotide and you just keep doing it, then as a consequence, you will encode this type of serine, leucine, threonine, alanine, leucine, serine types of amino acid out of this the gene sequence. However, for some reason, if the reading frame was like starting from the second nucleotide of this region, then you will have this type of reading frame. Then totally the amino acid identity will be different. So that's the concept of reading frame, which like more intuitively to our common routine life, what we can compare this reading frame is when you try to read a sentence, which is very comparable, similar to the organization scheme of the codon on the messenger RNA coding gene. So that I try to read this: the red dog ate the cat in one sentence, but all these alphabets are organized in three alphabets, so that then the whole sentence makes sense. So the red dog ate the cat, just like the case of three nucleotide forming one specific codon. Right. However, if a frame shift occurs, now you diagnose what happens in this particular occasion of frame shift, you try to read this information. Formerly, originally in the sentence, it was no problem for you to read and then understand at the same time. The red dog ate the cat, fine. But you try this same sentence, but something happened. So the red dog got the cat, kind of, it doesn't make any sense at all of sentence. What happened? Why? It's all because here you can easily find out, oh, this E has been deleted out, so this is a deletion, and as a consequence, still the codon, or here this the vocabulary, the word that positioned the previous to this event of deletion still retain its original identity, but afterwards all the codons has been disrupted, so now it's just became whole garbage. It's an example of frame shift of your original reading frame. Then it will be the similar occasion in the event of this point mutation deletion in your messenger RNA protein coding DNA sequence. Here, the similar thing happened. So original sentence, the red dog ate the cat, but frame shift, another type of frame shift, will totally also made this whole sentence is like impossible to read and make sense. The Rex dog got the cat. What is this? What happened? Because of this X has been inserted. Again, it caused a frame shift. Now, getting back to the original, the definition of the frame, the reading frame disruption, what was it about? Deletions or insertion of one, here in the example of this single nucleotide insertion or deletion, but it can also cause the whole this disruption in the two nucleotide. Scale. Why? Because after all, one codon consists of 3 nucleotides. So you try this. You add a 2 nucleotide, or you try to take out 2 nucleotides, then the same thing will happen. What about the 3 nucleotide level? If you try to add 3 nucleotide insertion, or if you try to delete the 3 nucleotide at a time. So let me show you what happened if you try to delete the entire 3 nucleotide. Then you're gonna miss this red. However, still overall, instead of the red dog ate the cat, but still it makes sense. You can still read the dog ate the cat. This is exactly what's going to happen usually, in not every time, but usually, if you delete 3 nucleotides at a time, then the chances are the identity and the function of the resulting the gene product as a protein is probably going to be saved. Okay, so that's what why, because the reading frame, in this case, the reading frame is still preserved. And adding things also, if you try to add another vocabulary, for example, if you try to add HOT 3 nucleotide, 3 alphabet, then what happens is that the red hot dog ate the cat. So although it is different, somewhat different from the original sentence's meaning, but still overall you kind of understand what it means. And then similarly, same thing with the chances are. Once again, it's not guaranteed in every different cases of this insertion or deletion of a 3 nucleotide will not have any serious consequences on the function of the protein, but mostly in this case 3 nucleotide levels of a change alteration will save this whole reading frame, but not in the scale of 1 or 2 nucleotides, because then it will disrupt, definitely it will disrupt the whole order of reading this whole nucleotide afterwards. Okay. Okay, so that's the type of the definition of the types of a mutation, and also we can classify the mutation by their consequence. Okay, so either as a result of a deletion or insertion or anything, if you had this whole the transcription, the no proteins, virtually no sensible proteins are generated due to this, then we call such type of mutation as a nonsense mutation. To be more specific, nonsense mutation happens when the stop codons are usually, if this is original messenger RNA gene, you have codons, but then these stop codons like UGA or UAG or UAA, either one of these three stop is located at the very last position of your whole codons of the messenger RNA. However, due to this frame shift, then chances are this stop codon with this random combination is a chance event. Now, this has been this stop codon. Now what happened is, as a consequence, even if you have this messenger RNA transcribed, you get to produce. Even with this type of mutation, you still make the messenger RNA, but this messenger RNA, as a consequence, will not be able to make any protein, because the translation will stop here only after making 2 amino acids. That's nothing. So that type of mutation, now we are viewing this mutation from the angle of the consequence, right? Not the cause of the type of mutation, just like as we seen, like is it the deletion or substitution or the insertion, but this type of so usually this nonsense mutation occur as a consequence of a frame shift. But it's not something that always the frame shift is the only cause for this nonsense mutation. No, it is not. I mean, something else can happen. So whenever there is a like abnormal stop codon has been generated due to whatever reason, but mostly those is we call as a premature stop codon. This premature stop codon, which generates like in the middle or at the early position of this whole amino acid codons and in a given messenger RNA, then we call that as a premature stop codon, because the stop codon should have been there at the very last position. Then we call it as a nonsense mutation, because the protein product will be very, very short. Okay, so virtually it's nothing. Another type of mutation from by the the consequence is called a missense mutation. Now here, you don't have any premature stop codon, but the product contains something incorrect as compared with your original. What you meant by this incorrect codons are included in this missense mutation. So as a consequence, usually, then protein is not entirely identical to the original version of the protein. So in that case, we call it as a missense mutation. Yeah. Whether this incorrect protein, how much incorrect, is actually what matters. Is it only the case of some kind of due to this missense mutation, you have some only a few amino acids have been altered, but still you survive because overall your protein function is not going to be changed that much, because after all your protein is consists of so many different kind of amino acids. So as long as you get to produce overall similar protein of similar functions, then you will be okay. And actually that is actually has been the one of the major driving force of this diversification of whole gene function through this type of mutation. Although along the past, some of those unfortunate victims of this mutation will get to inherit some incorrect proteins of the inferior function or even no function at all. So then the carrier of that mutation would have suffered the consequences. So that's all kind of a tough love. So anyway, so this when the proteins are still made, but that proteins are kind of incorrect compared with the original version, then we call that as a missense mutation. Okay. And another type of mutation from the angle of consequence is called a silent mutation. So sometimes you have this very lucky break. So, but mostly these silent mutations are generated by the substitutions. So that this substitution is the only mutation that does not generate what? The frame shift. So this frame shift is such a big deal in that regard. So even if you have the change in the nucleotide level, but sometimes you don't have any actual amino acid identity change, or you have a little of this change in amino acids, some kind of identities, but still, like I just mentioned a minute ago, you don't have any functional the compensation. What do I mean compensation? Functional sacrifice is too much. So in terms of function, doesn't overall affect too much. Why does that happen? Because one reason is the codon redundancy. Remember, you have only 20 different amino acids that need to incorporate in this protein building translation process. However, what's the maximum codon possible can you generate is actually 64 possible codon, but you subtract 3 stop codon, but still you have 61 available combination of this nucleotide codon. So that means definitely, obviously, you have more than one combination of the nucleotide, which point out one single amino acids. That's what we call it codon redundancy. Codon redundancy. as we have seen in actual codon table, more than one, usually more than one nucleotide codon specify the same amino acid. So even if you have this substitution, especially the third position, you have a 3 nucleotide codon, position 1, 2, 3. Then, as we have seen in the, just go back and then look up the codon table once again, then you will realize that usually the codon number 3 is the one that actually has a higher degree of freedom, or you can have whatever you want as long as you have this codon number 1 and 2 in nucleotide number 1 and 2 in a given codon. So actually is something that it is solid, fixed. Then the third position nucleotide can be anything, is usual kind of observation, general observation. So if this position of nucleotide has been substituted with something else, usually it wouldn't really matter at all. Okay. The second possibility is, even if you have this change in actual amino acid identity, but still this amino acid is in terms of its property, because out of these 20 different type of amino acid, you can group these all 20 different amino acids into kind of several different types of amino acid group, and those different amino acids within the same property group will actually influence as a similar like a function generator. Okay. So if you were lucky enough to have this type of amino acid change in from the same kind of amino acid in its property into another such similar kind of similar property of amino acid, then overall the protein shape, this all protein function will be determined by, once again, the shape, the structure. As long as you don't have any this whole big alteration in the protein structure, you will be okay. So those type of different types of mutation has been summarized in this. So even at the point mutation level, there are 3 different: silent, nonsense, and missense mutation, and what they are has been so again summarized as a kind of a conclusion of type of different mutation. So yeah, although we can have a ton of different things I can talk about this whole mutation, but yeah, that's not necessary at this level. So let's jump into the real life example of I'm going to pick up, for some reason, 3 different cases of this mutation. Okay, so first one is that's probably one of the most famous and frequently talked about in general biology course. It's called sickle cell anemia. That's been already mentioned at least a couple of times, even in this particular lecture. The sickle cell anemia, from its cause, it is a base substitution mutation, sickle cell anemia. But then probably your immediate reaction was, oh, that's good, because you said base substitution usually would be kind of a really mild, benign mutation, so it's not going to affect that much. But then why do you have this really grave consequence of this sickle cell anemia? Everybody knows that how serious it is, the sickle cell anemia, the condition is. Let's find out what happens, what kind of a really bad break you have in this sickle cell anemia case, even with this single base substitution. After all, base substitution means one nucleotide has been gone, but instead of that guy, somebody else has been sitting in over here. Hey, I'm here. It's just like, so what's going to affect that much? So this is substitution is in hemoglobin, as we have seen already earlier. The overall hemoglobin protein consists of two identical alpha chain protein, two alpha protein, and two beta. Combined all together, this 4 subunit, and the one functional protein of hemoglobin, which functions as the oxygen carrier. Then we usually describe such this sickle cell anemia mutation in this the beta subunit generating the protein gene is that this A has been altered into T, the nucleotide. Okay. So it's a substitution. A became changed into T, but then as a consequence of what happened is this E6V. What is this? Here E is, although like I said earlier, briefly mentioned that although the different amino acid we usually describe in the 3 nucleotide, like a like glycine. The full name is glycine, but we only just use a 3, the first alphabet, to designate glycine amino acids, and then so the all other 20 different amino acids. But that's even that using 3 letters is too much usually to those who are in this field. So it's more preferred convention of using the single letter convention notation. So E is a glutamic acid and V is valine. So that means E6V means glutamic acid, which was original amino acid, the identity. What does this number stands for? The position 6, the 6th amino acid was glutamic acid of all the entire hemoglobin beta chain protein, which consists of several hundred amino acids. Okay? More than 100, like, I guess it's a kind of a couple of, it was 300 level. I didn't quite remember accurately, but it consists of many amino acids. But so it's a very early part of this protein, the position 6 amino acid, position 6, which was glutamic acid, the E, has been changed into valine, another type of amino acid. That's what it means, E6V. And it's all caused by this single nucleotide conversion from A to T. So it is a substitution. Then, kind of without knowing anything, any more detailed information, probably your reaction would be, oh, so no big deal. You have this amino acid just conversion, but this original amino acid, glutamic acid, somebody else is like taking the role of this original glutamic acid. So what can possibly go wrong? But really seriously, something happened. And as a consequence, the protein folding to create proper structure has been totally went wrong. So created its incorrectly folded hemoglobin beta protein. Okay, and then those will accumulate not only beta chain, but it also affected the entire whole hemoglobin protein consisting of 4 subunits, 2 of which are an alpha chain and 2 of them are beta chains. Totally the structure has been changed, and then they keep piling up, like have this accumulated in the red blood cell with the wrong structure. So if you try to illustrate in graphically, normal hemoglobin, as you can see over here, 2 yellow alpha chain, 2 blue beta chain, whatever they are, or the color could be changed into. This is normal, the ball-like shape of hemoglobin proteins. But then with this one single amino acid change on that particular position, what happens is they have totally different. Original shape of protein was the cutie small ball-like structure, but now they have this whole clumped, this wrong structure of hemoglobin proteins got all connected one another, and then have this clumping and then settle down at the sink. It is not being solubilized in the body soup, and then accumulate in your red blood cell is what happens. And actually this is really not only in this case of the sickle cell anemia mutation in hemoglobin, but this clumping, protein clumping of wrong structured. So actually that observed in many different occasions. Example is diabetes. It's actually one major cause of this diabetes is one particular some proteins are their structures are altered, and the clumping of the structures is accumulated. And Parkinson's disease. Also, and Alzheimer's, and mad cow disease, to name only a few. So it's really the deadly, and what a kind of all these deadly, I would say, although some of them are not really like a life-threatening, all of them are actually life-threatening, the mutation based disease. They are all pointing out the same direction toward at the same moon there. So protein structure alteration, and as a consequence, it is wrong structured proteins are being clumped together and then piled up in the cell. So this poor red blood cell with this sickle cell anemia mutation, even they have the wrong shape. So normally this red blood shape is like a little bit of the nice little this donut shape, but without having any the central hole. This red blood cell shape become this shape. That's why it is a sickle shaped. Why? Because the red blood cell contains the majority of what's a mature red blood cell. Mature red blood cell doesn't even have any nucleus, and this cell dump everything but keep the hemoglobin protein, because knowing that okay, my only duty, my only sacred duty is carrying the oxygen. So for that matter, I will keep only the hemoglobin protein needed for that function, and everything else is just like a moot point. So I'm not going to worry anything about it, and that's kind of as a fate red blood cell exactly that. But then if that hemoglobin protein generated was just a wrong structure, accumulated clumped proteins piled up inside of the cell, then as a consequence the whole shape of actually the container, uh, which is a red blood cell, is also altered. And if you are kind of curious about why this mutation will result in this protein clumping, which was not seen in the so-called normal, I wouldn't say normal, it's a wild type, is usual the way of describing the kind of original version, unmutated version of any this gene. It's a wild type versus this mutant, and it's because in the wild type the protein foldings are like throughout the day. It's a consequence of a whole long period of evolution, so you get to have this particular shape of protein structure out of certain arrangement of amino acid sequences. So that's how you obtained this hemoglobin protein of a present function. But when you have like this glutamic acid at the position 6, actually what it is in glutamic acid is charged amino acid, which means during this protein folding, this charged amino acid will hate just the other way around, have no problem of face to water at all. I'm not afraid of water, so I will be brave enough to be positioned on the protein surface. Okay, whatever this water molecule, come on, I can handle, is what's happening. So based upon that, it is one of the such like the epicenter, a core of such protein structure building. But then when you have this altered, that position of amino acid, glutamic acid, it's changed into something else like valine. Valine is hydrophobic. Amino acid, which means I don't like to face water, so I will do everything in my power. I will do everything to avoid contacting with the water. So usually around any hydrophobic amino acid, you get to actually put the hydrophobic amino acid inside, so that based upon that total overall scheme of body plan of protein folding is made. So as a consequence, you have accidentally this hydrophobic residue is exposed. So, and then you don't like, nobody likes to see that. So what's the one quick solution of this, solving this exposed hydrophobic amino acid? Then you have this hydrophobic, try to connect themselves through this hydrophobic residue so that they can hide the area, the surface of the hydrophobic residue to be exposed to the external water, which is full of water. That's why you get to create this whole like a train connecting event, like a behavior like this clumping of proteins. And that's why you also get to observe such a similar clumping event in other the abnormalities, like did I say abnormality? Like mutations, like diabetes. Same thing happened. Then Parkinson's disease and Alzheimer's and the medical diseases. It's all based upon this protein structure building. So as a result, you have this sickle-shaped red blood cell and the functions. This With this type of red blood cell, your original function of carrying oxygen would be seriously challenged. So, so if you have, like, if you are homozygous, for this mutation, in other words, you have this chromosome, one homologous copy of a chromosome with the, let's say this is the position of your hemoglobin beta chain gene, and you do have a counterpart of your homologous, another homologous chromosome, which also at the same position, this beta gene, but both of them have been the mutated mutant. Then it is homozygous for this sickle cell mutation. Then all the products that the red blood cell will all be sickle shaped. Okay. Then you cannot carry the oxygen properly. Then if you don't have this proper oxygen carrying function, then it's called anemia, okay? Like lack of blood. Because no matter how much actual blood you have, in what matters in your blood is that, functionally speaking, is the red blood cell to provide the oxygen supply to every different part of your body. But that they cannot function properly, then it is as good as like you are experiencing blood shortage. That's why it can be called as an anemia. But then this anemia will affect all kind of other the secondary consequences, because if you don't have any oxygen supply, then you will have so many different kind of other functions also not being able to perform proper function. So then not only this sickle cell disease itself per se, but you will have so many different multiple disease symptoms will you have, right? So that type of the thing is called pleiotropy. Like one single gene alteration will have so many multiples of different traits. So one gene's alteration, one gene will affect the several different traits. Then we call such event as a pleiotropic effect. So in this particular case of this sickle cell anemia, or as a consequence, you have so many different functional consequences. On the other hand, so this is called the pleiotropy. One gene affecting multiple traits. On the other hand, the reverse, like kind of opposite is you have only single trait, single characters, skin color or anything, but that one trait is determined by many different genes. In this case, we call it as a polygenic inheritance. And surprisingly, actually, so many characters, you know the difference by now, the difference between character and traits. character is a big heading under which several different alternative versions of the traits exist. Yeah. So you can say this is character or the trait, whichever you feel like to. But anyway, the thing is, a single trait or character is determined by multiple genes, then we call it as a polygenic inheritance, which is exactly the opposite case of pleiotropy. And many different, like in eye color, skin color is one such a good example of this polygenic inheritance. And another thing is human height and intelligence, and there are so many actually traits that are made as consequences of this, the inputs of many different genes. And the one particular distinct characteristics of any trait determined by this polygenic multiple genes is it's not like a digital, like you have one or this, that or that kind of alternative trait, but the available traits are usually this like analog type of whole different array of distributions. You will easily notice that in the case of human height as a trait, like given in a population, in a given population, you have a small sample group of which extremely tall height and rather very short height, but the majority you have this average. Then it's not like you have one tall and one short, like only two alternative, but you have so many the variables can be observed within such character. That's a very distinct, typical characteristics of the genes, the character determined by this polygenic, the genes, polygenic, following the polygenic inheritance is. And we can easily, the human skin color can also be seen exactly the same type of such characteristics too. And getting back to the sickle cell anemia, it is a recessive mutation, okay, which means heterozygous. Having one gene is wild type and another gene is a sickle cell mutation is at least outside the phenotypically as a kind of a normal. Yeah. This heterozygous individual carrying only one mutation in one of the two homologous chromosome locus should no problem in maintaining the normal life, unless the individual decide to live in like a high altitude place like a Himalaya or Everest, like the location having very short supply of oxygen. So normally it wouldn't really affect. So it is a mutation, a recessive mutation. Why is it once again is sickle cell anemia a recessive mutation? What did I say about this in determining whether a certain mutation is going to be recessive or dominant? It's all about the function. If that, as a result of the mutation, if that protein is going to lose the function entirely, the lack of function, then is going to be a recessive, because wild type copy that's been kind of traditionally has been providing the normal homage, the expected function is able to cover this the missing function due to this mutation, and then in that case that mutation is a recessive, right? So same thing here. That's why in heterozygous individual with sickle cell mutation should no problem in maintaining normal life. Another interesting about things about this sickle cell anemia is, yeah, it's heterozygous individual has no the realistic problem of dealing with their normal life, but still it carries a kind of one copy of this beta hemoglobin gene is like functionally it's null, does not function. So it's not really, although overall it's okay, but still it's kind of you have two kidneys, and you miss one kidney function, but still one kidney on another the other kidney can provide the function, but still you are not kind of feel comfortable about it. Yeah, I'm carrying only one kidney. Okay, kind of. But here in this case of a sickle cell anemia mutation, you have actually advantages. Selective advantages. What do you mean selective advantage? In terms of a natural selection. Natural selection against this malaria infection. Yeah, there is a reason why this sickle cell mutation has such a the resistance against the malaria infection. I'd be glad to unfold the reason, the molecular mechanism why is that. But actually in the past several years ago in my class of this understanding life science, I actually took some time of in and took some time to explain the mechanism, but I decided not to ever since then. It's too much for those, especially those who are non-majors, so forget about it. Just like accept as a fact. Yeah, this heterozygous individual of sickle cell mutation is more advantageous in fighting against this malaria infection. So obviously those individuals will have a much higher selective advantage in the area where in the area only this malaria infection has been traditionally become problematic. Usually this temperate region or cold region where this malaria carrying mosquitoes are not there, you have no such advantages, but certain region, okay, has a problem of this sickle cell. So this sickle cell traits, this is a pink area, the red is pinkish area, mainly in the central Africa and some of these Southeast Asia and also Middle East area where malaria, here this green area, okay, indicated historical distribution of this malaria. So there is no coincidence why these two areas of these distributions overlap, because those areas where the heterozygous individual can have a better the survivability, and there is a reason why even you have this the allele. In terms of this allele, these two big A indicate this the dominant allele of so-called wild type. Wild type alleles of, like, so as a diploid you get to carry two such alleles as a wild type individual, no mutation, and a heterozygous individual having one wild type and one sickle cell anemia mutation. This is a heterozygous allele, and this is a homozygous recessive even though this homozygous recessive allele, yeah, will be much stronger in resisting the malaria, but it has a very difficult, like they usually cannot survive because of this whole the defects caused by this mutation. If that is the case, and in the population, this defective allele will be gradually over the throughout the course of the natural selection, evolution through natural selection, will be selected out. So it will soon disappear over time in a given population. But still, especially the frequency of this recessive allele is very high, unusually very high. Guess what? In this area, yeah. Why? Because in this form, if you happen to have this kind of a combination of being heterozygous, then you have a selective advantage in the natural selection process. That's why you get to keep this allele. So, in other words, if you examine the frequency, how much in terms of a proportion, how much of this allele, like a wild type allele of the beta hemoglobin gene versus the sickle cell mutant version of the allele is actually similar to a trait, but as a two individual haploid gamete, allele represent different version of this a character. We have defined the allele earlier already. So this allele, in everywhere else, if you try to count the frequency of this allele, then this sickle cell mutant allele will be extremely low, and that's what you have expected. Of course, it will be selected out. But in this Africa region, the reason, due to the reason that I have just explained, still the frequency of this mutant allele is unusually high. Is this all connected observation? The related observation is this. Okay. So why is the sickle cell allele recessive over the wild type allele? I have already explained. Okay. And by the same logic, even those kind of mutations are like alteration and some changes, usually the function as a consequence of the protein. Then why some others are dominant? If that mutation result in the gain of new function, or interfering existing function, then you will have the case of dominant mutation. So getting back to this sickle cell anemia mutation, instead of having this sickle cell mutation resulting in simple just piling up, so whenever you have this wild type gene, that wild type gene can form the normal functioning version of this hemoglobin protein. Instead, if due to the mutation of this sickle cell anemia somehow affect the formation of the normal structure of hemoglobin protein, that is kind of imaginarily possible, then suddenly this sickle cell disease mutation will become the dominant mutation due to this region. Okay. Now, the second case of this mutation is a cystic fibrosis. So if the sickle cell anemia was a consequence of this point mutation, which is a point substitution, which did not. Okay. So there, it did not have any frame shift. So that's where this is interesting things can come about it, because normally if you have this mutation which does not involve any frame shift, then you are going to be okay. But in this rare occasion of a sickle cell anemia, then even if it is a non-frame shift mutation, you will have such a serious consequence. Now, the second example that we are going to look at is a cystic fibrosis, which is actually really high in frequency in the Western, like a Caucasian population. So we usually don't have the Asian. As an Asian, we don't really have to worry too much about this cystic fibrosis. The incidence is extremely low. But for some reason, those the Caucasian population is unusually very high, and it is a consequence of the deletion, nucleotide deletion, but 3 nucleotide deletion at a time. So once again, is a frame shift. The reading frame is preserved, but still it has such a serious consequence. It is once again the pleiotrophic, in that it's a very serious disease affecting in so many different areas. The consequence of this cystic fibrosis 3 nucleotide deletion is in the protein CFTR, shortened or version of cystic fibrosis transmembrane conductance regulator. It is a protein. So I have just pronounced once the full name. So from now on, I'm also going to call it as a simply shortened form of CFTR, just enough. It is a protein. It is a membrane protein. It's a membrane transporter. What does it transport? Ion transport, something like a sodium ion or chloride ion. Types of these ions are being transported through this CFTR protein. So these CFTR proteins are everywhere in our cell, on the surface, on the cell membrane, and doing this transporting job. Okay, and the way we describe this particular mutation of cystic fibrosis is in this way. This is called a delta, Greek capital delta. So it is the same thing as a D, stands for deletion. So delta F508. Here F is another amino acid. This is a phenylalanine. So number 508 sounds like it is a really huge protein. It is, yes. So this CFTR protein is a big protein, and out of this position 508, that original phenylalanine amino acid is missing due to the deletion of 3 nucleotides corresponding to that phenylalanine. Okay. So, like I said, CFTR functions as an ion transporter in the cell membrane, like this. And so the importance of this CFTR is, due to this function, now we can maintain the liquid, the water outside of this all our organ and tissues. And so, like constantly flowing stream, this, thanks to the function of this CFTR, will maintain this kind of a body liquid flow. Okay. And that is extremely important in reducing the chance of the infection. So if, for example, in our respiratory the tract, outside of this our respiratory tract, we do have a constant flow of water, okay, and on top of on those surface of tissues we have all kind of the hairy like cilia structure, waving its movement function. So that's how we kind of shed off any potential infectant sources. But then when you have this deletion, even though you get to keep the overall the reading frame of the protein, but it results in the incorrectly folded protein CFTR. Yeah. And because of that, any incorrectly folded protein is not allowed to escape. So remember, it is a membrane protein, right? It's a membrane protein that is destined to be exported to the, so it is this CFTR protein will is the fate of CFTR protein is going to undergo this exocytosis to finally end up on the cell membrane, right? In other words, all those proteins that are destined to be exported through this exocytosis will initially go into the rough area, rough endoplasmic reticulum, and inside of which, as I described earlier, inside of which one is putting this carbohydrate, the glycosylation. Two, another one is protein folding. Okay, two very important functions will be carried out. Okay. But then incorrectly, if due to this missing amino acid, if the protein is going to be incorrectly folded, then that incorrect structure of the protein will not be allowed to escape and then shipped into the next destination, which is Golgi. Instead, it is all degraded, destroyed actively within this rough endoplasmic reticulum. So all these things is actually connected, linked to the earlier part of the lecture of that what we have seen as a eukaryotic the organelle function, which is still a part of our final exam coverage, right? So did it or did it not? I don't know, I forgot. Was the final exam coverage was like only the protein I was right. Yeah, it is still a part of this final exam coverage. So, and due to that, so you don't have any this liquid flowing function. It's like a compromised liquid, at least this function. Then this thick mucus, due to that, this thick mucus will build up in all those respiratory tract, including the lung area. But then it will make those area much more vulnerable to the infection. This is what has been described in this cartoon. Okay. And once again, it is a recessive mutation. Then you can easily guess why it is a recessive mutation, because the consequence of a mutation of this CFTL is the simple loss of function, loss. That can be compensated by the functioning copy of a wild type gene. Right? And like I said, it rather has such an unusual high frequency of the incidence in the especially Northern European heritage, which represent large portion of this Caucasian heritage in these days. And actually, that is the reason why in every biology textbook, this particular CFTR mutation is mentioned in general biology, because all these general biology textbooks were all made in that region. They still are the ones dominate this particular branch of science. That's why. And last example of this three mutation is called lactase persistence, which is not really that serious. It is not life-threatening mutation, so we can lighten up ourselves in following this instance. It is also a mutation caused by a base substitution. But once again, a very peculiar thing about this mutation is it is not on the coding region. It is not on the protein coding region, but rather is on the regulatory area of that protein coding gene. Yeah. So anyway, what is lactase persistence? It is about lactase, an enzyme which is involved in the digesting the dairy products. Okay. So this lactase is needed for digesting the lactose, the milk, the sugar present in the milk. Okay. So the lactase will cut off this chemical bond. The lactose is the disaccharide, okay, just like sucrose, and we have in order to metabolize, we have to cut this the disaccharide into individual component of a monosaccharide, and that's what this lactase does. Okay. So obviously, when we are in our development, when we are at the earliest stage of our development, like when in our infancy, when we're a baby, definitely we need the function. We have to depend heavily on the function of a lactase, because the mother's milk is the primary, or in most of the cases, the only source of the nutrition. Then the sugar present in those mother's milk, also cow's milk too, basically all of them are lactose. We don't have any immediately available glucose or anything like that, so that we can go on the respiration, aerobic respiration. So lactase is extremely important, but only when we are at very early in our development, like baby and infants. As we grow, as we mature, then now we begin to manipulate other real food sources, including bap and gogi and banchan. So we don't really need that much of this lactase, unless you are heavily the kind of a person really crazy about ice cream, like me. So that's the reason actually the humans are programmed to have this lactase expression. What do you mean expression of lactase? The transcription. So RNA making process, those expression of lactase will go down. We are already programmed to reduce, okay, cut it down. As you become older and older, the expression of lactase will. Then it might actually even, we may even reach to the stage of like completely shutting off this gene expression as we grow older. That's how we are. But then mutation occurs. So it is a mutation carried in some small population of people, but it's very high in, once again, it's a Caucasian lineage. So these people have this particular mutation, and as a consequence, the expression, the lactase expression continues even when they mature, even when they became older. And what's the nature of this mutation? So these individuals have no problem about. Actually, there is a reason why when you were young you didn't have any problem, but the it's called lactose intolerance. This symptom will develop as you grow older, like my wife. So in these days, my wife cannot really comfortably consume as much dairy product as I do. So when we were very early, like early stage of our marriage, we used to like 3 or 4 days out of and 7 days. We used to actually to stay up all night. I mean, finishing the whole bucket of ice cream over there when we were in the United States, and then we didn't have it. Both of us didn't have any problem. But nowadays, no. I am the only one still who can do, and sometimes I do still actually. She cannot do this because whenever she does, she will have this kind of like uncomfortable the gas accumulated in her like the intestine. It's due to because that you cannot digest this lactose, then your microbes in your intestine, your microorganism, the microbes will do this instead of you, and then as a consequence they will generate some their own unique the end product, metabolic product, which will obviously create some kind of a gas products. That's why you feel uncomfortable, but that's it, just uncomfortable thing. It doesn't really affect your lifespan or any health other than this being uncomfortable feeling. And but some people, maybe I carry this kind of mutation, don't have any problem. But that mutation is a point mutation. Fine, it's a point mutation, but not in the protein coding region of that lactase, but the regulatory area, or in particular the enhancer. What is the enhancer? If this is a coding gene produced messenger RNA, this is a lactase, then the regulatory region including there is a promoter, right? But also you have other also regulatory element, which is the enhancer. So the promoter region, RNA polymerase RP will bind, but the enhancer region there is what transcription factor. Both of them are necessary for this lactase expression, right? And this particular enhancer region there is a point mutation one. So this is C residue at this particular enhancer region will be the will be responsible for this lactose intolerance. But some people carry this point mutation, which make this conversion of a C to T, then it will become lactose tolerant, like me. Yeah? But another very interesting, surprising thing is the distance between actual lactase gene, and this particular point of enhancer region, which is 14,000 nucleotides apart. Are you kidding me? This 14,000? Such a long distance, still this particular area can function as a kind of a control regulatory element? Yes. So a lot of times actually that's what it is. So this is what about what it is about this lactose intolerance and lactose tolerance differences. On the other hand, actually, if you have the other way around the mutation, which allows that you are not able to utilize this lactose sugar even when you are a baby. That particular lactose intolerance will become very serious defect. That's kind of another type of a genetic mutation, which is not this kind of a lactase in this, so intolerance related. I mean, this lactase in, what was it? I forgot. Why did I do? I forgot the original lactase persistence. Sorry about that. So this is about the lactase persistence, but intrinsic lactose intolerance symptom can be caused by other types of mutation on the area of this gene. And if that was the case in babies, then they have a really a serious problem in that they cannot consume mothers and any other types of milk products which are essential for their stage of development. Okay, so that's another story. So with that, I think, oh, okay, so it is dominant mutation. Then you guess why it could be a dominant mutation, because it is a gain of a new kind of function, right? Yes. And so probably perhaps those ancestors who remained at this particular painting on the walls of this cave are the ones who first developed this mutation. So this frequency of the mutation, lactase persistence, is very high in Caucasian populations, as I said. And on the other hand, like Asians, like us, and those Native Americans are very low in frequencies. So this indicates these relative frequencies where the darker you have in blue color, the frequencies of this lactase persistence you get to see in high. One last comment about this is the population who have this over area. For example, like Mongolians. If you knew already, like these people are also heavily on the dairy products of sheep milks are the main source of their nutrition. But as like a similar ethnic group of as us, they then what happened? Did they develop high frequencies of this lactase persistence mutation? No, it turns out that not. No, it turns out not. But still, they can tolerate such lactose even at the later age, an older age, is because of this totally different reasons. Because of this microbes in their guts. So it once again shows the importance of this, the microorganism that we're carrying inside of us. So they somehow get to carry some specific microorganism who can easily digest this lactose, but still does not cause any uncomfortable gas buildup. That's how they can manage this. Okay, so with this I can finish this the first part of this lecture. This time I get to finish I get to use about 1 hour and 30 minutes. So let me think about for a moment whether should I actually include another chapter of a lecture or simply finish at this point and then say good luck on your final exam. If I decide not to include any additional sessions, so I will just say the my final comment about this lecture over here. So okay, so this is what it is about chapter 5, and good luck on your final exam. If not, I will I will add some more additional material in the second session, and then see you there.